So it's indeed a pleasure to now call Dr. Gilles Salvat, who is Managing Director General of the Research and Reference Division, Scientific Director of Animal Health and Welfare at ANSES. Dr. Gilles is the former Director of the ANSES Laboratory on Avian, Swine and Fish Diseases and Foodborne Zoonosis. He is the Scientific Director for Animal Health and Welfare since August 2011 and became Managing Director General of Research and Reference Division in February 2018, this year. He will be presenting on genome-wide identification of host-segregating epidemiological markers for source attribution with Campylobacter jejuni. Thank you.
Thank you. So thank you so much to invite me for this presentation. It's another example of what we could do with WGS, and it's a comparison of what we have before and what we could do now. Because source attribution in Campylobacter jejuni is of great concern. I will take the same picture as Jürgen. But as you can see, all the Campylobacter are not coming from Denmark. And even if France declares only about 6,000 Campylobacter per year, the real estimation is about 500,000, more than 500,000 Campylobacter in France, regarding the statistic we have from hospitals and so on. So the real part and the real burden of Campylobacteriosis in public health is much more important, even in France, than it was considered previously. And it's the same, of course, for Europe, using the same way of declaration and estimation. There is many potential sources of Campylobacter, and the most common admitted sources is chicken and poultry in general. And there is many publication assessing the role of chicken and poultry products in the contamination of humans, with two main risk factors: consumption of undercooked broiler meat and cross-contamination during preparation of broiler meat. But we have this project regarding the identification of potential sources, because poultry, even in the country where poultry is considered as the major source of Campylobacter, mainly 20% of the other sources are not identified. So we try to make some investigation about which are the cause of the real sources of Campylobacter in human, and we have the hypothesis that it may be ruminants, it may be poultry, of course, and environment with its largest exception with wild birds and mollusks and so on. So what are the methods which are able to make source attribution in Campylobacter? The gold standard method at the moment is MLST, multi-locus sequence typing, which is methods which consider 7 housekeeping genes, the sequence of 7 housekeeping gene and allele sequence of this housekeeping gene, and by comparison of this different housekeeping gene sequence, you make clone complex, what we call ST for sequence type, and this could be used, but the limitation of these methods is that you only sequence 7 genes. Whole genome sequencing, MLST, will be another promising way to make source attribution regarding that you will sequence all the genome. And first, we published in 2016 a method. It was made by, this work was made by Amandine Tepo, which was who was a PhD student in our lab. together with Sam Shepard from University of Bath. And they found 15 genes which are associated with host-segregating epidemiological markers for source attribution in Campylobacter. And we want to compare these methods with ST complex. But potentially, we have a good improvement of the sensibility in probabilistic assignment of sources of Campylobacter with this method. Another method, and this is the last one we compare, was CGF14, which is comparative genomic fingerprinting, which is an old method now, published in 2012, and who is supposed to amplify 40 ancestral genes of Campylobacter. And the comparison is made on a binary principle. So the gene is present or not, and it makes a combination of 0 and 1 for 40 gene. But the kind of question is that method interesting for source attribution? So we compare on different sources of Campylobacter strain. We compare these three methods with high number of strains from poultry, even from beef and sheep meat, less strain from pets because it's more difficult to have because the surveillance of pets is not so good. We have some strain from the environment from our colleague from IFREMER, French Institute for Sea Studies, study of sea, sorry. And we have some human strains from the National Reference Center in Bordeaux Hospital. And we compare strain from human origin from 2009 and 2015. So we compare some different source and different number of strains. And what we analyze is the results with bionumerics for and probe-MLST for MLST allele designation with big SDB for complete whole genome sequencing, and we make the probabilistic assignment with the software structure. What we found with that, first from our French cases from 2009, yeah, without using the source of PET, we found that, and it was a surprise because we do the same job with English strain with the Bath University and Swansea University, and they found that the majority of the cases were due to chicken. But we found in 2009 that equally part of chicken and ruminants are responsible for Campylobacteriosis in France, especially using host-segregating markers, which seems to be the most efficient methods for source attribution. When we added PET strain, we found that PET strains is also with, especially with host-segregating markers, so whole genome sequencing, PETs is a non-neglectable source of Campylobacteriosis in France. When we do the same with the strains isolated in 2015, you have higher implication of chicken compared with ruminants, and you still have high implication, not so negligible implication of PETs in the source attribution. But in both cases, host-segregating marker is the best way statistically to attribute for source attribution for Campylobacteriosis. as a conclusion, we could see that there is difference between those three methods, and that the whole genome sequencing methods were the best one. I'm not sure that you will have such a good source attribution for other strains than other species than Campylobacter jejuni. Probably it's these 15 host-segregating genes are specific of course, they are specific of Campylobacter jejuni, but I'm not sure that you could do the same with the same methods and the same methodology with Salmonella typhimurium, for instance, which is more wide present in different animal species. But we could say that these methods, or segregating methods, is the best method at the moment for source attribution for Campylobacter, and that you have to do this source attribution on a regular basis. You couldn't say, yeah, in France, half of the Campylobacteriosis are definitively associated with beef, because as you could see, comparing 2009 and 2015, that there is a big difference. And probably there is evolution in the contamination of raw products. And why ruminants in France? Probably because in France, we use minced beef meat undercooked. And that's probably, we know that the contamination of ruminants is very lower than chicken, because chicken is about 80% of the chicken poultry meat is contaminated with low amounts, but with Campylobacter. But there is certainly an effect of the raw mincemeat from beef mincemeat as a vector of due to the low cooking or raw meat use in France. Environment is not a negligible sources, and pet implication have to be more investigated, probably by doing specific studies, epidemiological studies on contamination of pets, which may be very important, especially for young children. Chicken is also in France highly implicated in human contamination, like in other countries, but there is a significant role of ruminant reservoir. And it reminds me that Santé Publique France, which is the public health agency in France, in 2004, they published a case control study on Campylobacteriosis, and they found that one of the risk factors was the consumption of raw minced beef meat. And in 2004, when they published that, we said, yeah, that must be a mistake, because it's chicken. And probably we have the proof that it was not a mistake, and that epidemiology is also a good thing for source attribution. Thank you for your attention.
Thank you so much.